Thank you for joining us today. Today we have Dr. Sergey Plis presenting on machine learning for predictive neuroimaging. Dr. Plis is an associate professor in computer science at Georgia State and the director of engineering at the Center for Translational Research in Neuroimaging Data Science. He's also a co-founder, president, and research chief at Reasonate.ai, a natural language processing startup. As such, let's go ahead and welcome Dr. Plis.
Thank you, Anthony, and hi everyone. So I wanted to give an overview of what my group is doing, has done in this area in machine learning for predictive neuroimaging. But then I realized that it's too much, so don't treat it as a complete overview of our work, but treat it as some several milestones that are in the focus of what we're doing now. And of course, I am the presenter, but there are many people behind that work. Some of them are below the slides, some of them will appear on other slides, and some of them will not appear as the pictures, but I will hopefully mention them by name. But science is collaborative, so it's many people in this work. Since it's kind of a longish talk, I wanted to talk for 70 minutes, but Anthony didn't allow me. But still, so here's what to expect. Motivation, background. I will try to stay within 45 minutes, don't worry. I was just trying to scare you. I will talk about semantic segmentation and our recent results there. I will talk about interpretable machine learning. That's where predictive machine learning is interesting. I will talk about causal learning, our old results, or some of our new results, and I will not talk about decentralized learning because I will run out of time about there. But I forgot to remove that bullet point. So let's first talk about what motivates our work. We want to non-invasively figure out what's going on in the world by measuring something about the world. Instead of going around and poking with a stick and putting gear and going into jungle and actually going to expeditions, we want to do remote sensing, and one of the biomedical remote sensing MRI, a couple of modalities as you see on the screen, but there are other modalities for the brain, and there are other data collection mechanisms which are non-invasive, which are not, which are more passive and observational. So if we do that, for example, for the brain, you see that cyst there in the patient brain. So it is nice that the scanning has been done. I don't know if the patient already had some symptoms and that's why they got scanned, or as it's often happens, somebody got signed up for a study and then accidentally people who scanned the person figured out that there is a problem. But the goal of neuroimaging and brain imaging is we want to collect observational data and understand the disorder, what is happening there. For example, if we knew there are symptoms in behavioral changes, then we want to understand the mechanism, what is happening. So the natural response to this desire of drawing inference from data is improve our data collection mechanisms, and so we do, right? That's how we arrived at the age of big data in 2000s. And then we didn't know what to do with it. But anyways, that's the next slide. So let's collect more and more data. And it feels wonderful, right? The more we know about the brain, for example, or the more we know and measure about everything, there is a semantic difference here. The more we measure, right? The more we measure does not immediately translate into the more we know, because now we have huge multidimensional, at the tiniest possible, at the current technology state, resolution data. But then how to process that data, how to see through that data. Okay, if you have to work only with one person worth of data, maybe that's possible to sift through all the possible dimensions, but usually we want to see populations, effects of in populations, and so on. So it's very hard. So we have a trade-off or misbalance there. On one hand, we still believe, I think that's the current state, that the more data we collect about the world or the brain, the more we will learn. But at the same time, the more data we collect, the less we can learn, because the data is hard to convert to knowledge and extract the essence of it. What is the meaning there? So the dream is, and all opinions are mine, but I hope it represents an alignment, at least with people who work with me, is to obtain an automatic solution that goes away, loops through the data, comes back, and helps us interpret what's going on in the world. And that thing, or that interpreter, okay, that could be a person, right? Often a specialist, but specialists are expensive, non-scalable, and also they saturate at some point. So we want an algorithm. In this talk, in my work, we want an algorithm. Robot is a cute representation of an algorithm, but it's think of it as some form of computational model. So now think about neuroimaging. So it studies the brain, produces immense amount of data. Well, you already figured. Data comes in various forms, each with their strengths and weaknesses, and our goal is understanding how the brain works and breaks, because we study mental disorder where the brain is arguably already broken, right? There is a problem. So the brain is, what is it, right? Where to stop, at which resolution? If we're collecting fMRI data, functional magnetic resonance imaging, that I will talk on at the next slide, I couldn't align those slides properly, then what in the brain is the part that matter? At which resolution should we stop? Is it neuron to neuron talking to each other? Is it voxel to voxel, like an arbitrary cut region of neurons? Is it a region to region? In this work, we kind of converged that it is region to region, but it's not an easy process of going from voxel representation of these masses of neurons to regions, which regions in the brain are active. But another problem or another interest is that it's not just the problem of figuring out which regions in the brain are meaningful, coherent agents. That means which voxels do we group together. The problem is that those agents, or the regions, we call them regions of interest, right? Or if you use a different estimation mechanism, you can call them intrinsic networks. But they're active constantly, and their activity is changing. And often we're interested not only in estimating which regions are coherently active, but we're also, or mostly interested in the spatiotemporal pattern, where and when in the brain something interesting was happening. So that's the problem, and the problem that we want to figure out from the data. Functional MRI is one modality that measures how the brain works. So the brain, well, from the name, it's brain function. It measures blood oxygenation level dependent response, produces a 4-dimensional movie, a cube changing in time. It's relatively well localized, but sampling rate is low, and actually, because it measures metabolic activity, even if the sampling rate is high, still it's very blurry. The changes are not as quick. And then magnetoencephalography and the sister modality, electroencephalography, at least thanks to Maxwell equations, it measures magnetic field and the strength, instant reflection of underlying activity. But the weaknesses, which are arguable. Maybe some people believe there are no weaknesses, but on certain spatial location, at least you have to solve an inverse problem in order to get to a spatial location. So another background, well, the point is, even though those are totally different things, and magnetoencephalography, electroencephalography, and fMRI all are actually related by through Maxwell equations. But let's not get there. Let's assume we're measuring magnetic field directly in MEG, and we're measuring metabolic rate indirectly in fMRI. But the assumption is that both of those things, or both of those signals, are produced by the same activity. It's neural activity. So we have two views of the same underlying thing. That's now to the mathematical models that will pop up in one of the sections. Bayesian neural networks. One of the goals is to estimate, use predictive machine learning, or use machine learning to split the brain into regions. Another one is to analyze the dynamics and find out where and when things are happening. And another one is to find out causal relationships between brain regions, which regions directly cause changes in activity in other regions. For that, that's the third part that I will highlight. For that, we are using graphical model representation. But it's a representation. We use causal graphical models. In this representation, the whole joint distribution of, think of X on the screen. My mouse is visible, yes. Think of X on the screen as collection of nodes on the left, which represent the brain, locations in the brain. And this is the joint distribution. You can factor it in multiple ways, but if you factor it in, say, one true factorization of families, conditional distribution, then you get sparse representation of the joint probability distribution, because otherwise you have to represent each pairwise interaction or each multi-way interaction if you want. Because if you have a two parents, amygdala, say, and hypothalamus, and hippocampus is the child, this is a probability distribution, conditional probability distribution here. That is not just pairwise, but it's a three-dimensional conditional probability distribution. Okay, so now we're ready. So let's predict something about voxels, and let's split the brain in a relatively simple task called semantic segmentation. We want to take state-of-the-art in terms of accuracy and in terms of widespread use algorithm or a tool called FreeSurfer that finds regions of the brain in subject space. Given a volume, a three-dimensional cube, FreeSurfer spins the wheels for a few hours. Used to be 8. I think it's still about 8 hours, and segments the brain into gray, white matter, and then gray matter can be colored by FreeSurfer as well. Semantically, we all have those, again, arguably unique patterns of the brain folds, similar to fingerprints, but FreeSurfer manages to find the regions that are specific to each subject, and this cannot be done by color of the tissue, because tissue is just gray levels, and it's more or less uniform everywhere, the patterns. Yet classical signal processing algorithms of FreeSurfer and the art of the creators, they managed to figure out all of the semantical details. So, but 8 hours is too long, especially with big data set, 30,000 subjects, and you need to process all of this relatively quickly. You don't want to wait 8 times 30,000, so that's too long. We want to take a model, deep learning model, neural network in this case, that would take the brain as an input and produce the output. Two tasks. One is just gray and white matter segmentation. If you're familiar with the problem, you can also think, or you can use this for brain masking or brain extraction. And another one is semantic segmentation, now beyond the gray and white matter into the semantic regions of the brain. Classically, already, given the dates, but it was fresh at the time, people came up with an architecture of a convolutional neural network that is called UNet because of this U-shaped structure here of encoder and decoder of convolution. And the thing with the brain, it's a three-dimensional structure. And let me skip some slides to show you the problem. So when it's three-dimensional structure, and you call a human to label the voxels manually, the task is for each voxel, it's a prediction task or classification task, to each voxel in the 256 cube volume, assign a value from the class. For gray white matter segmentation, it's either gray, white, or outside the brain, right? 3-class problem. But for semantic, that's many class problem. So when a human approaches the task, given a digital brain, how would you do it? You do it by slices, right? You open one slice on the screen and another slice on the screen. The problem with this is human, this is ground truth, and this is the model that I will tell you about. But why did the human make this mistake? I don't think it was carelessness. I think it's more about slices. When you look slice by slice, in order to retain consistency, you have to look in all of the planes of projections, or at least in many planes of projections. And that's it's already tedious task. This is even more tedious. So the premise of our work, of this work, and of many people in the field, is that a model can look at 3D patches or a 3D brain, the whole brain together. And in 3D, relationships are clearer simultaneously. Well, like the model is not limited by binocular vision, as we are sort of plane of view. It can sense, quote unquote, everywhere simultaneously. So, but if we do that, and we form a volumetric, that's the term for it, UNet model, then the model is 23,500 parameters in size. 23,000 parameters in size is about 2 gigabytes. And now if we need to train it on a lot of data, on multiple GPUs, on multiple nodes on the cluster, or across, even worse, across the research centers, then each iteration will have to pass around 2 gigabytes of data. That's a lot. And then also, if I want to share that model with you, you need to download 2 gigabytes of data. Of course, 5G, fast, I can do it, but maybe it may not fit on your phone as easily, right? So what we came up with back in 2016 and published in 2017, that was Alex's work. We came up with a different alternative architecture that doesn't use UNet shape and doesn't use encoders and pooling architecture, but uses dilation and uses sort of isomorphic layers. They're all the same. The advantages, it's still volumetric model. The advantage is that the number of parameters at the same accuracy as UNet is giving is compared to 23,000 to 23 million. That wasn't 1,000 years, 23.5 million. And 72,000, but the difference is 2 gigabytes in size and 600 kilobytes in size. At the same accuracy, they're really not real measurable advantage is in using MeshNet, Alex's model versus UNet. They're about the same. We can get MeshNet perform better if we add better than UNet if we add more parameters, etc. That the interesting observation, and this applies to all of the deep learning. It's more of deep learning applications to neuroimaging than advertisement or whatever results of our MeshNet. But the interesting thing that even when free surfer, we train it on free surfer data, not on human labeled data. Human labeled data is hardly available and very expensive to generate. And we show here that free surfer makes mistakes sometimes. It incorrectly extracts white matter and gray matter. But MeshNet, our model, others will do. It's a general deep learning property. They generalize over non-systematic errors like that. So even on the same data set, the ground truth where free surfer makes mistakes, here, as you see on the left in this column, MeshNet is able to generalize over free surfer's mistakes. But it was at the time making some different mistakes. It's not ideal. Now we have something much better. Another thing is in many applications, not just in brain imaging, but not just in neuroimaging, in other radiography applications, it's required to look at multiple modalities in order to make a clinical decision. It's already hard to analyze one three-dimensional cube, but when you have three cubes, it's looking 3 slices at a time, looking at all the projections of those cubes at the same time is very hard. But the models in deep learning in our model can just take 3 volumes at the same time of all modalities and volumetrically look at it. The problem at the time, and still to many models, and now we're going to some unpublished results, is that if you work on the whole brain, where the advantage should be the greatest, especially when you're labeling, say, areas of the brain. When you're looking through a small aperture, it's very hard to first know, are you in the front of the brain? Are you in the back of the brain? You need to learn a lot of details in order to be able to predict that. But the large brains, they don't fit in the memory of modern GPUs. The models that process, maybe now A100 GPU has 40 gigs. Some models do fit there. Another version of that GPU has 80 gig of memory. And so people can train very large models within that constraint or not constrained anymore. But in 2017, we were unable to do that, and many people still don't have access to those devices. What we used to do, and still many people are doing, is looking at small cubes, chopping the brain into very small cubes, and applying the model to those cubes, and then stitching together the results to get better results. But recently we realized that our compact and portable model also has an advantage because it lacks some architectural disabilities, let me say, of UNet. It can be run even on a small 10 gigabyte graphical card, GTX 280, for example, just for the sake of argument, on full brain. And when it runs on full brain, it simultaneously views the whole brain, and apologies for anthropomorphizing the model. I don't mean it actually, is conscious, only slightly maybe. And because it's tiny and because it sees the whole brain, it's an advantage. It can be made even tinier with even fewer parameters, high accuracy. It can be run in browser now with good accuracy. And when it runs, let me show you an example how it looks. So we have brainchop.org that is running. This one is just a demo that is running. I want to show you. so not used to talking to silence. Yeah. Okay. If you have any questions while I'm messing here with the details, I would gladly take them. So this is now a full brain segmentation model, gray-white matter segmentation, running in my browser. The only like confession is that this is Apple Silicon MacBook, not MacBook Air, but otherwise it runs on many others, although it may fail because of memory. So we're testing it. So this is the result that it produces in real time of gray-white matter segmentation. And I can tell you that it's pretty good. Okay. And with the overlap, you can save it. So that's the advantage of this model. The slides, I don't know if we will make them available, but at least from the video brainshop.org, you can test the subcube model there. That definitely works, and the experimental model is different, and the code and everything is available. It's open source. So that was about using predicting machine learning for splitting the brain into region. Now, what about the dynamics? And for the dynamics, the goals are slightly different. This is Mahfouz, a student in our group. It's mostly his work, but we have other people working in the field, Noah and Osman as well. But we have a paper under review that I will tell you about now. So the desire is to work with dynamical data, resting state data. I don't know if I need to introduce to this small crowd what resting state data is, but basically a person is in the scanner instructed to do nothing, just not fall asleep. Sometimes eyes open, sometimes eyes closed. So different people don't think the same thoughts, and the signals are not alignable in principle. There is no common task that connects the subjects. So we want to know what's going on, and that's we're getting to that little robot reading a book. The premise of this work is, can we train a model to predict whether a person in the scanner, well, who we collected data from, has a diagnosed mental disorder or doesn't have a diagnosed mental disorder? We call one control group, another one, say, schizophrenia. We're doing that to different disorders, to 3, and I will talk about them. And we're interested in interpretability of the world, not the model. There is a lot of talk right now about model interpretability, trustworthiness, alignment problems, and things like that. But for our work, we want to learn from the models, not about their inner workings. We want to learn from the model about what in the data made them generate a certain prediction. So if the model is very good at predicting, say, or separating schizophrenia versus healthy control, we want to learn from the model where it is looking at and when in terms of the timing of the signal, because we believe that not all of that resting state data is neurotypical, say, or has disorder. It's probably there are spatiotemporal patterns that are schizophrenia specific and Alzheimer's disorder specific, and so on. So to understand our difference in this desire from many people in model interpretability. It's called model interpretability usually, but I dropped the model interpretability because this is not my goal to interpret the model. Imagine I'm working with you on some problem, and when you solve the problem, I want to learn about the problem, not about your feelings when you were doing it, or your inner workings, or like, did you drink coffee or not? So it's similar. The whole process is in this picture, and again, the Qt robot, hopefully you view it as Qt the same way as I do. We construct a model. We train this model to work on the time courses from the data. Each subject has a time course from each corresponding intrinsic network or brain region for simplicity. Don't want to go into detail. There are spatial components with their corresponding time courses that we obtained using independent component analysis, but that's not the key here. And then the problem in this work is that when you have spatial-temporal patterns, you are in the curse of dimensionality mode. You have very high dimensional data, and usually we don't have that many subjects to train a deep learning model on high dimensional data. So what we did, and that's not topic of this discussion, we first pre-trained the model. And then we trained the model, tuned this model, this pre-trained on healthy controls, human connectome projects, large data set. We pre-trained it, and then separately, independently trained or fine-tuned on autism spectral disorder dataset to distinguish autism patients from healthy controls or controls. Schizophrenia separately, Alzheimer's disorder separately. Binary classification for all of them. And then we use the interpretability to figure out where is the model looking at and when. So this is time dimension and a few examples of few subjects. But you can say, well, logistic regression gives us a very interpretable results, or even like regression. Why don't we use it? Why do we need all of that deep learning and all of this trouble with pre-training? So if we apply to the same data, dynamic data, because we're interested in interpreting spatial-temporal patterns, we are not interested in creating features that we know are already predictive, and we know that classical machine learning algorithms will work. So we apply it to the raw data, the data that I showed you. Well, raw in a sense that we don't construct the features, but it's still processed dynamical data. The red line or the dark brown, depending on how you see it, is indicating a chance level 0.5%, 0.5 area under the curve of ROC curve. And we see that for autism, all of the methods are mostly random. For schizophrenia, this is the number of subjects per class on the x-axis. For schizophrenia, slight improvements, but 0.6 is not good. So those models don't work. Well, our initial thought was, why don't we use recurrent neural networks? They're so good at time series data. And yes, recurrent neural networks actually can work on this data if pre-trained well or if there is enough data. But the problem is here. The problem is called vanishing saliency. And vanishing saliency is that if this is synthetic data on the left that we've generated. I can do this, I can do that. So this is our ground truth. That's where in time the signal that we encoded is encoding the class. Class 1 and class 0 distributions are different. Recurrent neural networks, LSTM, supposedly the best ones of them, they only looking at the last time point. They're giving us interpretability. They're always pointing to the last time point, and then saliency maps are vanishing. We constructed a different model where we fixed that problem of vanishing saliency interpretability, and that's where many people involved in coming up with this architecture. It trains well, it pre-trains well. We constructed specific pre-training method based on self-supervised contrastive learning. And our model points us exactly where we should be looking at, where the underlying signal is. Then the model itself is powerful, but with pre-training, it's statistically significantly better at when the data is small. So when you have small data sets, the red and the light red again, you can see that we beat classical methods, although this data is very small for deep learning. The next time when people tell you that they want to reject your work because you have a small sample and you're using deep learning, point them to our work. And we found interesting patterns in the following sense. We found that, we know a lot about dynamics already. We know that functional network connectivities of those respective disorders, they have specific shape. But the way we look at this functional network connectivities, it's by compressing all of the time courses of all of the regions into one matrix or one graph. With our searchlight, the model looks at the data and highlights the part of the data that are predictive of the class. We take only 5%. Here's how: 5%, 10%, 20%, 30%, just an example mask of how much of the data is actually used. We don't use any of the 10, those are just for illustration. So 5% of the data only already highlights the same relationships that we know are present in autism, schizophrenia, and Alzheimer's. So the model is actually pointing at the right thing. Then we went further and asked the question, is the model always looking in particular place in time? And we asked it because of things like here, right? There are multiple places in time. This is only time. X-axis is time. And we have patients and controls for each of the three disorders that I mentioned. Because it has some temporal pattern, it's not like, okay, I'm looking here, I'm looking there. We ask the question of differences between healthy controls, the way the model looks at healthy controls, and at patients. And we devised a metric, because this is kind of messy. We devised a metric of peakiness, of how compact, because that's what's important for us, how localized in time this predictions of the model are, or explanations, let's call them explanations. The model is telling us, look in this time window, look in that time window, that is disorder specific. And we found statistical significant differences of this, the way, using our metric based on Earth mover distance, of the way patients and controls are viewed by the model. Like, for example, you already can see here in schizophrenia, patients kind of come up with the term healthy everywhere. It's more spread versus, healthy controls are more spread and patients are more peaky. The model is actually looking at specific points. So similar difference is for Alzheimer's disorder, but for autism spectrum disorder, it's all spread. There are no significant differences, or equally spread, I would say. So that's interpretability. How are we on time? We started later, but it's 6:49. Anthony says, what should I do? Were you thinking I will finish at 6:45?
So is this the last section?
Well, but look at this. It's okay. Yeah. It is the last section, but we'll skip the other one. But it's a little involved. It'll take me 20 minutes to go through.
Okay. I think it'd probably be all right if we go ahead and call it.
Yeah, then let me do some. So this work is done. Many parts of this work and many parts of the work that I didn't show are done by this group of people. and I still have a postdoc position and possibly another PhD student position. So if you know someone who's looking, let them send me an email. And questions? Interesting. Image froze there.
So I liked how in the interpretability, you looked at the distribution of the importance. Are there other works that have done similar things that compare the degree of localization of importance between?
Yeah, I haven't seen any of the work like that. Yeah, that is looking at this. Especially, I have not seen, besides maybe two papers, other than us interested in interpretability of dynamics of dynamical data, because I can only guess because of the difficulty on training on high-dimensional data. Dynamical data is high-dimensional, and in order to be interpreted each point in time and space, you need to train on that dimension. You can't compress it, right? Yeah, I don't know, like that's my guess, but not too much work there, and we follow the literature pretty closely.
Okay, awesome. And so I guess you compare the degree of peakedness between controls and individuals with neurological disorders. What conclusions can you draw from the statistically significant differences there? Just that the model treated them differently, or can other additional conclusions be drawn?
So I can talk to schizophrenia part, right? So let's look just on schizophrenia here. And for people who are familiar, this is FBurn dataset. Of course, there is uncertainty, and what I say is not true for each row in this dataset. And this is just an example of subjects. For example, if you look at the first row, the red and the blue look the same. But then the further you go, it's patients controls are more just widespread, looking everywhere, versus patients more localized. So the hypothesis of the more localized patients for schizophrenia is that the expression of disorder may not be continuous. Like it's a psychotic disorder, and we even call them psychotic episodes, right? Even the people who have the disorder, they don't constantly dwell in psychotic state. So my hypothesis is, or our hypothesis is, that when you're in the scanner during that resting state scan, it's also most of the time you're going through a normal state, but sometimes you're kind of going into psychotic attractor, one or another, and that's the timing of it is different for each patient and different within that scanning interval. That's at least the working hypothesis for now. Like, why would the brain be always psychotic, right? And imagine even in epilepsy, when we know what is happening, the seizures are infrequent. It's not like constant seizures. So similarly to that, it's an analogy, of course. I don't want to pretend that this is exactly the same thing, so don't overinterpret what I'm saying.
Okay, that's very interesting. Thank you.
Yeah, thanks for listening. Thanks for joining today, and thanks for the chance to highlight some of our work.
Definitely. Are there any more questions?
Okay.
Thank you, Dr. Plis. This is very interesting, and we appreciate your presentation. We'll be posting it on YouTube as well. And I know there is a number of people that weren't able to attend today, but we're interested in watching it online afterwards.
Sure. Thanks for inviting me, Anthony. I appreciate that. It's my pleasure.
Thanks so much, and have a great day. Thanks. And thank you, everyone, for joining.